Hello everyone, today we have the second lab in the series of pGLO transformation and GFP expression. And today we're going to talk about the results of previous seeding and also proceed with the GFP expression. Following overnight incubation, we have bacteria grown on plates. And if you look at the LB agar plate with bacteria not transformed on them, you could see them growing as lawns or smur. Now look at the LB ampicillin plate with the non-transformed bacteria, which did not grow. The LB ampicillin plate with transformed bacteria grew as single colonies, and the same happened for the LB ampicillin rubinose plate, where the transformed bacteria grew as single separate colonies. Now we are going to observe them under the UV light. Please note that observation of bacteria under the UV light needs the special protection, in our case it's face shield. So when we turn on the UV lamp, you can see that the bacteria on LB ampicillin rubinose plate glow green, while other bacteria don't glow green because they don't have arabinose in medium. It is because the gene of GFP is under arabinose operon promoter. The arabinose induces the expression of GFP. We proceed with the experiment and we are going to inoculate LB broth medium with a single colony of transformed bacteria. To do so, you need the inoculation loop. Pick up a single colony that contains transformed bacteria. Do not dip it into the agar, just touch it softly. Then transfer to the tube and make sure that you mix it well. Because our medium contains ampicillin and arabinose, it will induce the expression of GFP in transformed bacteria. For negative control, you will be growing the non-transformed bacteria in usual liquidous LB broth. We are going to use 2 ml of LB broth and flat bottom 14 ml tubes to make sure that our bacteria grow nicely. The third tube is the duplicate for our transformed bacteria. Take the plate with the transformed bacteria growing on LB ampicillin arabinose, take one colony and transfer it to the tube. Immerse the loop inside the LB broth, mix it, and waste the loop. Now it is ready to be placed in the 32 degrees shaken incubator. You are going to incubate it overnight. We set the temperature of incubation to 32 degrees to make sure that our bacteria do not overgrow in the tubes with medium. After transferring the tubes into an incubator, set up the rotations per minute. And in this case, as you can see, it is 150 rotations per minute. Make sure that the temperature is set to 32 degrees and press start. Because of the open frame construction of our incubator, the temperature will go easily below 32 degrees, so you can see 31.7 now, but of course it will go up.